Testing, testing. Excellent, good job. I've known Deepak now for 6 months, and he is a rather humble man. So indulge me while I talk to you a little bit about who he is and his background. So Deepak is a MD PhD by training, trained in medicine at the Mass General, did his fellowship at the Farber and spent some time at the Broad. Since that time, he moved to UT Southwestern, where he is associate professor in the Department of Medicine, also a member of the oncology division, and he is a UT Southwestern presidential scholar, as well as Ryan mentioned, distinguished chair, the Joseph Sambrick Chair. In other words, he has the type of academic career that I was not smart enough to have. So welcome to Deepak. I want to start at sort of a local level and then broaden out. And I wanted to, you know a little bit about our RBM 39 program. When I came to Recursion about a year ago and I started to learn about the program, I reached out to my network and said, I'd like to really sort of bounce this off of a real KOL. And person after person said, you really have to talk to Deepak. Deepak is sort of the father of RBM 39. But I guess sort of the first question that I'll ask, about our program. Could you talk to myself and the audience a bit about sort of the history of this target from in your lab and sort of where it's been and where it's going?
So. Sure. Well, first of all, I want to thank you for having me. It's really a treat to hear about this program and about this tremendous platform for phenotypic discovery. So maybe just by way of introduction, I can talk about a little bit on how we got into RBM 39. We never set out to study that, and our program is in some ways similar to what you guys are doing, but it's a single, I'm a single person. We don't have a giant platform. So, I trained as an internist and then as an oncologist, and I was a biochemist by training, and then I landed at the Broad Institute. And I wanted to go back. When I was at the Broad Institute, there was, kind of early days of RNA interference and nominating targets. I would watch people kind of get a list of targets, and they all had assigned some statistical value, and they would just kind of go down the list and look for which ones were druggable. So they'd be down to 10, 15, 20, looking for a kinase and looking for what's druggable. Because at the time, we had all these proteins that could be potential targets in disease, but no way to really target them with small molecules. And so I moved to Southwestern, actually, and Steve McKnight recruited me there, and he had really built a department around phenotypic discovery. And the idea was to start with a small molecule and then understand its target, and then by definition, it would be druggable. And so Steve really convinced me to build an entire program based solely on that. So we started to use kind of newer technologies that had come about, like click chemistry and forward genetics, to take small molecules, kind of orphan compounds, and try to understand how they were actually working. Because small molecules may be 400 or 500 Daltons, and as Laura just showed, it can land in all these different places in the cell, depending on concentration, into little pockets of proteins and affect function in a really precise way, some that you want on target and some that you don't. And so the challenge is kind of like a needle in a haystack. Can you take this small molecule where you have no idea how it works, you know that it's killing a cancer cell, and can you find out how that happens? And so what we've done over the years is to build genetic tools to basically give us a single mutation that takes us right to the key binding site of that molecule. And so that's what happened with this program. We built our first generation forward genetic system, and we started to put a lot of orphan compounds through. And one of those compounds was this compound that was discovered by Eisai Pharmaceuticals back in the late 90s. And that compound, we found mutations in RBM39 led to resistance. And as it turns out, this compound turned out to be a molecular glue, and RBM39 was recruited to an E3 ubiquitin ligase. So, we did really kind of deductive reductionist science to get to the mechanism for how that chemical targeted RBM39, which I'll say is, RBM39 would be a conventionally undruggable protein. And nobody, unless you're approaching it phenotypically, no team of medicinal chemists or structural biologists could imagine how you would get a small molecule to target RBM39. So that's one of the real benefits of phenotypic discovery with small molecules is you end up, finding things that people couldn't predict.
So if you look at the literature, RBM39 has been a target that's been around for a while. You discovered it, you in your lab. It's been brought into the clinic in a relatively narrow indication. It's been evaluated in AML and MDS. Could you talk about some of the learnings, and maybe the studies were not that positive, right? It clearly demonstrated target engagement, but it has not lived up to the expectations of when it came into the clinic as a very exciting target. Any thoughts? I mean, it's a complicated question, right? But any thoughts in terms of some of the, I don't want to say missteps, but the lack of prediction preclinically to what was seen in the clinic, and then I'll sort of talk about our approach.
Well, I think, drug discovery is like a checklist. You have to have all these various things checked off in order to have a positive result in the clinic. You have to know who to treat. You need to have the right small molecule. It needs to be bioavailable. And that's why you end up getting these kind of screening funnels. And I think Recursion is kind of turning that upside down. So it's not a funnel necessarily anymore. So in this case, this molecule was discovered and these trials were run in the late 90s, early 2000s without knowledge of the target. So if you don't know the mechanism, you have a lot of risk. You don't know if your compound's bioavailable. You don't have pharmacodynamic markers. You don't know if it's, like someone said, all bound to protein. Is it actually getting to the target? So I think this was, and most importantly, you don't know who to treat. I think, over the last 25 years, it's widely appreciated now, and we know this in the clinic, that, no one's cancer is alike, that lung cancer, any cancer, is a whole different set of diseases. And so you need to know exactly which patients are going to depend on a particular target. And if you imagine you have no idea, you're putting this molecule in a person and you don't really even know what you're targeting.
I mean, one of the things that struck me when I came to the company is, have we really uncovered novel biology around RBM39? Clearly there are indications that one could go after with a better molecule, and we believe we have a better molecule, but that's not what Recursion is trying to do, right? We're really trying to utilize our arbitrage to go after unique biology. Now, a lot of that we've talked about, but could you sort of comment on not only the approach, but some of the data that you've seen and we've talked about in the HR proficient solid tumors?
Well, I think that's a novel insight that your group has come up with. I think it's interesting. I just sent you this manuscript recently where, people had done phenotypic screens now with RBM39 degraders in PDX models, across multiple PDX models, and came to the same conclusion that your algorithms predicted. And so I do think that is the key element for that, is that, once you have, let's say, a small molecule targeting this now undruggable protein, is there a clinical problem that you can solve with that, and who are those people?
So let's pan out a bit. We've talked a bit about RBM39. I've looked at your work over the years, and your lab has evolved, right? And I think I would characterize Deepak's lab as somewhat atypical in the world of academic medicine. Could you just sort of talk to the audience a bit about how your lab has evolved and why it has evolved in terms of using a phenotypic approach or a phenomics-based approach? And perhaps the second part of the question is, why is it a novel approach? Why aren't more labs investing time and effort and thought into this sort of approach? So kind of
yeah, I mean
walk us through that.
that's a question I think about a lot, because actually phenotypic drug discovery is how drugs used to be discovered, how many of the drugs we use in the clinic were discovered. And in the last 25 years, it's really switched to a target-based approach. And that was for some of the reasons that the RBM39 degraders failed. We didn't know the target. Without knowing the target, it was really difficult to advance drugs that were discovered phenotypically. And as a result, it became expensive and uncertain. And now you can go with a target-based approach, and you have a precise protein, you know what protein you want to target, but you have an entirely new challenge, is how do you get a small molecule to target that protein? And as a consequence of that, the number of targets is actually really finite. So you have an intense amount of kind of competition on a small number of targets, and there's this vast amount of biologic space that's kind of unexplored. And I think phenotypic screening can now come back because things can be done in parallel. So you can look for compounds and targets and patients all at the same time. Whereas in the past, some of the most successful drugs, like lenalidomide, for instance, we were talking about, the phenotype was discovered in the clinic, 15 years into the process, and turns out to be a blockbuster drug, but that's a long time and a lot of uncertainty. So with the tools that you guys have and the tools that have come now, you can find the molecules, the targets, and potentially the patients much quicker, and start to explore this space. So we're not targeting the same proteins, over and over again. And I think the models for different companies have become kind of very risk averse, so that people are targeting the androgen receptor again in a slightly better way. And that can become, I think we're leaving a lot of potential clinical opportunities on the table by focusing on just a small number of targets.
One of the things, I've worked on probably 20, 30 different molecules in the clinic, and I've never worked on a molecule that did not demonstrate some degree of activity. But the challenge has always been trying to identify why patient X responded and patient Y responded, right? So it's my bias that it is as much about the target as equally to understanding the patient. And I wonder if you can sort of talk about the prioritization in your mind in the characteristics of what molecules should have when they come into clinic, and talk about sort of, I'll let you chat about that.
Well, I think, going back to patient selection again, how early in your clinical development pipeline can you see responses? How well can you predict who would respond? All of these things will really, as you, of course, as all of you know, as you move along in the process, it becomes much more expensive. The experiments are much more expensive, the trials. So I think having, and in addition to that, I think kind of pharmacodynamic markers. When the first RBM39 degraders in the 2000s were used, they were dosed at dose-limiting toxicities. So, having knowledge of the mechanism, and I think your pipeline here, if I could comment on it, can really nominate new targets. And then if you add on to that really rigorous kind of mechanistic mechanism of action and pharmacodynamic markers, you're poised to really explore explore this space.
So, talk to us a little bit about where you think we'll be in a few years from now. Do you expect this approach to have greater uptake? Do you still envision recursion as sort of leading the field, but a very small field? Talk to us about sort of not only in your view across other industry, across companies, but even talk about sort of your view academically. Do you see more folks, more labs embracing this approach, or do you think that it's simply going to be a matter of time where people will just follow, the leader?
Well, as you mentioned, my academic approach is really unique. Most academics focus on one disease or one problem, and we've decided to focus on, well, there's thousands of chemicals that kill cancer cells. Let's just find out how they all work. And once we know how they all work, we'll have a much larger toolkit for potentially treating disease. And I think that the academic model doesn't favor that kind of research, but I do think Chris showed this graph of the loss of efficiency. So how can we have so much technological advancement in chemistry, structure, cryo-EM, and still lose efficiency? And I think it's because we're way too focused on a narrow list of targets. And so I think Recursion's efforts to expand target space and to take those to the clinic quickly will potentially kind of turn the tide. I think academics oftentimes will follow, unfortunately, because of the academic model. When Recursion is successful, phenotypic screening will be back in favor academically too.
Because as you mentioned, right, it was right years ago, right. Phenotypic screening was the way to go, and you sort of talked about the evolution. Do you think that there's the sort of lack of embrace is an unfamiliarity with the technology? I know that a lot of when I speak to investigators about recursion, and I talk about our approaches, oftentimes, and these are very, very bright people, their eyes glaze over, right? Just talk to me about the target, talk to me about the population, talk to me about the trial. So do you think it's a bit of lack of understanding, which could be
maybe also talk about who else is targeting that protein?
Right, right, the other piece, right. So is it, again, I'm trying to, because this is important. There, a lot of people here, some of which have embraced technology and what we're doing, and others here that are appropriately skeptical. So, talk to us a little bit about the skepticism. And is it really going to be simply a matter of time between validating targets in the clinic, or do you think it's going to be something more than that?
Well, I think it's going to be about the beginnings of some clinical success following targets that are not conventional. I also think that right now you have KRAS as a target, clearly mutated, new compounds that hit there, and then you have a whole bunch of companies that are in kind of a fast follow, trying to get the better KRAS inhibitor, or we saw it with CDK4, CDK6 inhibitors. So I think Recursion has the challenge of kind of being the pioneer. The very first ones to describe all of your programs are likely first-in-class targets. And so I think I don't think right now there's been appetite in big pharma or small biotech to really have the courage to pursue these kind of phenotypic studies. So I think once you demonstrate their success, one, you're going to have a lot of fast follow, which I'm sure you're ready for. And two, I think you will open up that the phenotypic screening might be actually more productive, more profitable because you're in a novel target space and you're ahead of everyone.
So I'll finish up on this. Deepak is here on double duty. He flew back from Scotland to participate in a download day, and he's also meeting with our discovery team later today. We'll hear about partnerships that we are doing with companies. I just want to talk to you a bit about potential partnerships with Recursion and academic labs. You're here. We're doing work together. Where do you see the opportunity, sort of the complementary approach? What can you help us do, or any guidance that you can provide?
Yeah, I mean, for us, we have our own mini platform, where we take a small molecule where we have no idea where it goes in the cell to kill a cancer cell, and then our platform generates a result where it says it's going exactly to this place. And that mutation and that knowledge provides a tremendous asset to lead optimization and drug development, because it helps you pinpoint exactly what's an on-target effect. And so that's some place where we could help with compounds that you have, where you have some promising phenotypes, phenomics, but you don't know the exact mechanism. Because even though you want to move quickly, and with just 3,000 or 4,000 compounds quickly get to IND, you don't want to miss anything. The stakes are high. So all the knowledge you can have, cryo-EM, in vitro assays, all of the things that come with traditional target-based programs, you also can deploy with knowing the mechanism. That's one place we can help, and I would say that's the best place. The other ways is that for chemicals out there, maybe even in the public space or in our space, knowing who to treat, who would benefit.
So again, I want to thank Deepak for coming, spending time. I think he arrived late last night around 11 o'clock, but he looks fresh and bright-eyed. And thank you for his partnership and his guidance, and really thanks for the chat.
Thank you, David.
Thank you, David. Thank you, Deepak.